Hi everyone, welcome back to Food Feeds. In this video, we are going to talk about enzymes. This is one of the topic which is very important for most of the exams, and you can expect questions from this topic in almost all the exams like ICAR, JRF, CFTRI, GATE, FSO. So we will cover the basics today in this video. That is, firstly, we'll know why catalysis is essential for life, what are enzymes, why do we choose enzymes over the chemical catalyst. We'll take some of the real life examples of enzymes and also the main feature of enzyme, that is specificity. We'll discuss about it and also how these enzymes work. So let's start with the first question. Why catalysis is essential? So basically, what is catalysis? So it is a process of increasing the rate of chemical reaction by using a substance called catalyst. So catalyst is itself will not be consumed during the reaction. Rather, it helps to lower the activation energy that is required for the reaction. So any chemical reaction, when it is taking place, it requires certain amount of energy, and most of these chemical reaction in our body, it will need a lot of energy expenditure. So if catalysts are present and if there is a presence of enzymes, then these energy requirement will be lowered. So this will help us to carry out the reaction faster. So if we take the example in food industry for catalysis, it we can consider nickel. Nickel is used as a catalyst when you are converting or when you are hydrogenizing the vegetable oil to make margarine, right? So now we need to know why this whole process is important. Life to exist itself, we need basically two important things. That is self-replication, and also the second thing is the catalysis of chemical reaction needs to be done efficiently. So without catalysis, the reactions like even breaking down of sugar would take years. You might have seen that when sugar is kept in our kitchen, like in a box, nothing will happen to it for like years together, right? But as soon as it enters our body, it will be converted to energy. So this is possible because of the presence of enzymes in our body. So they basically just turn these reactions, which is a slow process, into a life-sustaining one. So in short, enzymes make the chemical reaction by easy by speeding up the slow reactions into a faster one. How? By lowering the energy required, which is called activation energy. So it lowers the energy requirement for the reaction to happen. Next, we will know what exactly are enzymes. So are they just proteins? Exactly. So enzymes are biological catalysts. Most of them are proteins, though there are some RNA which are not, but there are some RNAs called ribozymes which also act as enzymes. So they have some properties, like they are produced by living organism, they are protein in nature, they are unstable at a very high temperature or high heat. That is why most of the enzymes get destroyed at a high temperature, and that's why they are also called thermoliable. And they work in colloidal form in the inside the cells, and they are extremely specific, which means that each enzyme will recognize its own substrate. So, in short, these enzymes are specialized helpers of life's chemistry. So, next is that why do we choose enzymes over chemical catalysts? So why not just use chemical catalyst instead of enzymes? So, well, chemical catalysts, they often need very high temperature, they need a good amount of pressure, and also sometimes they need strong acids to work. But on enzymes, on the other hand, they can work at normal body temperature, normal pH, and they are very specific in nature. So there is one enzyme doing one job at a time, and it can speed up the reaction to million times faster, and this can be regulated or even inhibited. So, which helps us to control the complex pathway in our body. And that's why the nature always uses enzymes over the harsh chemicals. These chemical catalyst is mostly used by humans in the industry, right? But by nature itself, enzymes are the one which carry out the most of the chemical reactions. So now we'll look into few of the examples um, of enzymes that are in different industry, how they're used. So basically, medicine and pharma, there are drugs which are used as enzymes. You might have heard of glucose oxidase, which is used for diabetes testing. And in food industries, renin is used in cheese manufacturing, amylase in brewing, and protease in bakery. And in agriculture, they are used to improve the soil and even the crop processing. And one interesting thing is enzymes are also used in detergents. So the enzymes help to break down the stains that are formed on the clothes. So basically, every time we eat bread, we have wash the clothes or test sugar, enzymes are basically doing their work on them. So now let's explore the main feature of enzyme, that is enzyme specificity. So this is the one of the amazing thing about enzyme is their specificity. They choose exactly which reaction to catalyze. So it is basically the ability of each enzyme to recognize its own substrate, and they carry out a particular type of reaction. Each enzyme will have its own reaction, and there are different types of it. That is stereospecificity, reaction specificity, and substrate specificity. So we'll explore this one by one. So in case of stereospecificity, the enzyme will differentiate between isomers. We know isomers are the structures which have the same formula, but they have a different structure. So, for example, if it is following stereospecificity, the enzymes, then um, if you have to break the alpha glycosidic bond, only amylase will work on that. So, for example, if you have alpha glycosidic bond, and if we have cellulase in the same place, there is no reaction taking place. But if if we have cellulase, it will only break down the beta glycosidic bonds. It will not act upon alpha glycosidic bonds. So they are very specific that certain enzymes will only act on certain isomers. So this is about the stereospecificity, and there is also a reaction specificity. Here, there will be a same substrate, but on the same substrate, different reaction can take place. For example, if you consider protein, amino acid, it can either go transamination, where there is just a transfer of amine group from one compound to another, or there is oxidative deamination, where the amino is released. So there is the reaction, the enzyme will work on the same substrate, that is amino acid, but there will be different enzyme working on that substrate, which will give different results. Next one is substrate specificity. So there is absolute specificity where there is only one specific substrate. Like there is glucokinase, which will only act on glucose. Urease will only act on urea. So glucokinase cannot go and or act on any fat particle like lipids, right? So this enzyme will only work upon a specific substrate. So next is relative specificity. So here it acts on related compounds or specific groups. For example, if you have lipids, so this will contain ester bonds. So any lipase, any presence of lipids, lipase can work on it. Similarly, if there is a presence of peptide bonds, trypsin can work on it, breaking down the peptide bonds. So next is broad substrate specificity, which can act upon a wide range of closely related substrate. For example, glucose, fructose, mannose. They can be all acted upon by hexokinase. So how do enzymes work basically? So they are powerful because they speed up the reaction by lowering the activation energy. So if you can see the diagram here, usually, the required energy for a reaction to take place is higher without the presence of enzyme. But when there is the addition of enzyme or presence of enzyme, the activation energy, that is the energy required for the reaction to take place, is lowered. Hence, it will happen faster. So to explain these, how do enzyme work, there are two types of model, lock and key and induced fit model. So in case of lock and key, what happens is the enzyme and the substrate together are a perfect fit. Means the substrate has the shape exactly require and that shape is exactly perfect fit in the enzyme with the enzyme, which you can see on the left side of the image here. Whereas in case of induced fit, there is a the shape of the substrate is taken up by the enzyme to fit in the substrate so that it forms the enzyme substrate complex. So here E stands for enzyme, S for substrate. Then usually together they form enzyme substrate complex. And at the end, you get the enzyme and the product. P is for the product. So basically, enzyme is always present during the chemical reaction and it is just will lower the activation energy. It will not be used or utilized in the process. So you will get back the enzyme as it is along with a product formed. So there are two types of model, the lock and key and induced fit model. In lock and key, the substrate exactly fits to the space made by the enzyme. The structure is completely fitted into the enzyme structure, but to the active site mainly. But in case of induced fit, the substrate is as it is. Enzyme will make a shape so that it will adjust its shape in a way that substrate can come and form the enzyme substrate complex. So this is all about the basics of enzyme in this video. So further we will talk about other features of enzymes, what exactly is active site and different concepts of enzymes. So do subscribe the channel and hit the notification bell icon so that when I upload a new video, it will reach to you. Thank you.